Hello students, welcome to my channel. Today in this class we will learn about DNA microarray. What does it means? DNA microarray. Why we need this? Because in our human body all cells have same DNA, but their activation in every cell is not similar. Means all gene not compulsory active in all cell. Their different expression in different cells. Means gene activation level and activity is different in different cells. Means like if some gene active in liver cell, it doesn't mean that it also active in kidney cell. It can be active, but their expression is very low. That can be active more highly than liver. So their activity is differ from cell to cell, organ to organ, tissue to tissue. But all cell have all type of DNA, but expression can be on or off. So normal cell function and disease caused gene expression will be different. If there is cell which expressing some kind of cell, normally it working efficiently. But if there some cell is diseased, their expression could be stopped or could be even higher in rate than a normal cell. So we can compare both cell which is disease and which is normal one. So earlier when microarray was not invented, that time scientists could study only few gene at a time. But after microarray means nowadays thousands of gene can be studied simultaneously. Means thousands of gene can be studied at a time, at single time. So what is microarray? Now we should find out the answer of what is the microarray means. Microarray is a technique in which orderly arrangement of thousands of non-DNA sequence. Means there is arrangement of non-DNA sequences and which are arranged, number is so high, like more than 1,000 non-DNA sequences are arranged on the solid surface. So that non-DNA sequence is known as probe. Why probe? We'll see next upcoming session. Fixed on solid surface. What type of solid surface we use here? It could be glass slide, it could be silicon chip, it could be nylon membrane. Any one could be used in this technique. So what is the other name for this technique? Other name are gene chip, DNA chip, bioarray and gene array. You have to remember all these names because in exams, if you don't know all these names and question will use this term as the microarray. So you should not be confused because you know this technique in detail but you don't know other name, you cannot answer, you can be get confused. So you have to know all these names. What is the principle on the basis this technique is work? Principle is based on Watson and Crick base pairing. What is the Watson and Crick base pairing? Watson and Crick base pairing are like A will base pair with T, C will base pair with T, so G, and that is vice versa. What is A? What is T? What is C? What is G? A for adenosine, T for thymine, C for cytosine, G for guanine. These are the nucleotides, and that base pair with particular these two nucleotide, pyrimidine and purine. So pyrimidine will base pair with purine and purine with base pair with pyrimidine. So these, and there is also a rule that only A will make base pairing with T, not that T will base pair with C. No, never. If there is happening, so that will be considered as a disease. So what this base pairing done? There is should be complementary between DNA or RNA will get hybridized to probe DNA. What is probe DNA? Probe DNA are those DNA which we do place all 1,000 on the solid surface. That was the non-DNA sequence are the probe. And that if there is complementary in our sample, that will get hybridized with that probe. And that hybridization is follow the rule of Watson and Crick. A will bind with T, G will bind with C. This is the base pairing happen. So next, binding is detected using labels, fluorescent or radioactive. This kind of labeling will done for the detection, and these labeling are done with the sample we are using. Sample will label with these chemicals. Fluorescent are dyes, and radioisotopes, radioactive elements will get label to the nucleotides of our sample material. Expression profile, what does it means? Main use of DNA microarray का हम mainly use कहाँ, where we use mainly these DNA microarray? Measure presence of gene expression, means if there is expression happening or not, and if there is happening the gene expression, then what is the amount of that expression? These are measured as expression profile. And for this we use mRNA because DNA self is not considered a expression. Expression is done when DNA convert into the mRNA and mRNA convert into the protein. That will consider a expression. If DNA is converting into itself, this is not the expression. Expression will show their activity. What is the other use of DNA microarray? Other than this identifying the 1,000 of gene expression and all, what is the other use of microarray? Apart from the mRNA expression, microarray used to study DNA and protein interaction also can be studied with the help of DNA microarray. Means which type of protein? Protein which are interact with DNA, and with DNA which kind of protein will interact? Mostly the transcription factors will interact with the DNA. And another we can study alternative splicing. What is the alternative splicing? If there is 1, 2 and 3 intron are there in mRNA, then how they are splicing and how they are binding with each other. If we cutting from here, there will be 2 and 3 with together and 1 is separate out. If we cut from this by the process, transcription process, after post-transcriptional process, if we cut from here, we will get 1 and 2 with together and 3 will be separate. This is the alternative splicing. If we cut from this and both side, then we can find that can be all three can be separate out, and these three can be joined one first one intron. So this will make the genome expression different. So this is also a study with the help of microarray. And epigenetics. What is epigenetics? Other than DNA expression, what is other than DNA expression can participate in genetic expression, which is DNA methylation, where the methylation occur in the DNA on the chromosome will consider as epigenetic. That epigenetic changes also can study in microarray. DNA copy number variation. In different DNA, what is their copy number and what is the replication procedure, replication rate will be considered as the varied number of copies. And this is also studied with the help of microarray. Sequence polymorphism. What is the sequence polymorphism? Sequence polymorphism is morphology, means if DNA is for a like with a DNA which is working in ear and function is same, but what if their DNA of one man and second man is different? Function is same but sequence is different. That is also can be studied with the help of microarray. So, DNA probe in microarray. Which type of DNA probe? What is the probe? This is also we will learn in next class. Okay, bye-bye. Take care.